Hello and welcome to our latest video. In this video, we will show peptides technology development at Iktos focusing on three main points. New peptide fingerprint representations developed at Iktos that are more adapted to peptides. Prediction and generation of peptides taking into account more complex peptides. And some additional tools for peptides clustering, visualization and alignment that can be helpful when working on peptides. One of the challenges to model peptides is that peptides are very heterogeneous. They can have very different structures, different length, they can be linear or cyclic, and they can include natural amino acids as well as modified residues, or even other specific chemical components. At Iktos we can successfully process different kinds of peptides. We can deal with different length of peptides, natural and modified amino acids as well as linker and other chemical components that could enhance peptides' druggability. We can work with linear peptides, cyclic peptides including various type of cyclization, as well as multi-chain peptides. Let us move to the new peptides representations. Morgan fingerprints is a classical representation used for small molecules. It is a vector of fingerprint built from the atomic graph of the molecule. However, they do not seem to be suitable for peptides, as only a very large radius is able to encode the residue sequence. At Iktos, we implemented new fingerprints more suitable for peptide and applied machine learning models on it. By drawing inspiration from the Morgan algorithm that builds fingerprints from the molecular graph, we introduced new graph representations of peptides at the amino acid level that will be used instead of the molecular graph, to build fingerprints more adapted to peptides. We introduced different graphs at the amino acid level. The first one is the simple amino acid peptide graph. It is an amino acid level representation where each node of the graph corresponds to an amino acid. The second one is the backbone sidechain peptide graph. Each amino acid node is split into a backbone node and a sidechain node. In these graphs, edge type and stereochemistry is considered. Another important feature in the Morgan algorithm is the choice of invariants to define each node content in the graph. In our case, we choose two different invariants. The first one is the amino acid token or name that will be used to represent each node. The second one is the amino acid descriptors. Following user input, a list of descriptors is computed for each node of the graph. The descriptor values are then binned into intervals. Each type of graph is combined to a type of invariant and then converted to a fingerprint using the Morgan algorithm. The combination of the two kind of graph and the two types of invariants gives four new representations. AA tokens peptide fingerprints, BBSC tokens peptide fingerprints, AA descriptor peptide fingerprints and BBSC descriptor peptide fingerprints. We also defined another type of graph. It is a fragment level representation that is between the molecular and the amino acid level representation. We call it the pharmacophore graph. In this graph, the peptide is split into multiple fragment nodes following predefined functional groups like hydrophobic or hydrogen bond acceptor. The pharmacophore graph is then converted in a vectorial representation using the Morgan algorithm, considering as invariant the pharmacophore group name. This representation is also very useful for more classical small molecules. After defining the new representations, the next step is to compare them to classical representations. Morgan fingerprints on atomic graph and the descriptors. In order to do that, we run prediction experiments on a dataset of 189 small linear peptides with their measured target 1 and target 2 PIC50. Peptides of the dataset include modified amino acids and other specific components used to enhance peptide stability and permeability. The aim of this experiment was to predict activity on target 1 and selectivity on target 2. We used classical logistic regression model to compare the representations. We compared the representations on two different splits. First, the random split where data is randomly split into a train set and test set. Second, the stratified single linkage split that is more complex, where we try to put data of different distributions in the train set and the test set by using clustering. It is used to evaluate model generalizability. The plots shows the AUC results on the test set for the two targets. For target 1, on the random split, we can see that peptide fingerprints perform better than Morgan fingerprints and descriptors. On the stratified single linkage split, peptide fingerprints perform better than Morgan fingerprints and descriptors. We can see a significant gap between BBSC token representation and classical representation. For target 2, on the random split, peptide fingerprints and Morgan fingerprints have equivalent results. On the single linkage split, surprisingly descriptors alone perform better. However, for fingerprints, the one relying on descriptors invariance seems to achieve better performances. In conclusion, peptide fingerprints have promising results, especially when considering their generalizability. Indeed, experiments have proven that they generalize better than classical representations. An important feature that needs to be satisfied by the representations is the interpretability. When looking at the interpretability of the Morgan fingerprints on peptides, we find it limited for multiple reasons. As peptides are big molecules, there are too many invariants computed by the Morgan algorithm. The computed invariants by the Morgan algorithm don't encode the amino acid sequence, which leads to poor interpretability on peptides. This will not help the chemist to understand which part of the peptide is important to improve the wanted feature. On the other hand, peptide fingerprint interpretability is more representative for peptides to understand important amino acid sequence patterns for target activity. In the example below, the figure shows that the invariant 417 is important to enhance peptide activity, and this invariant represents the sequence L, G, W. So the chemists can know that this sequence is important in the peptide to have good activity values. For peptide generation, we adapted Iktos pipeline used for small molecules to peptides. The peptides generator generates peptides in PLN or protein line notation format. It is a textual format used to represent natural and unnatural peptides. PLNs are suitable for machine learning, hence our choice to use them for generation. An example of a peptide PLN is shown in green on the slide. The first step of the generation is to train a model on a large peptide database to learn the peptide PLN syntax. Then we launch a reinforcement training loop, where we first generate peptides using the model. Then the generated peptides are scored by the multi-objective reward function. This function can include different scores like machine learning model predicted scores, or other defined scores like similarity quality and confidence scores. We also defined checkers that will check that the selected peptides match defined criteria like pattern checker or PhysChem descriptors checkers. Then the weights of the model are adjusted to maximize the probability of generating peptides, similar to those maximizing the global score using a policy gradient algorithm. We launched a multi-parameter optimization using prediction model scores and other defined scores. We were able to generate more than 62,000 peptides satisfying constraints. That is a target activity score greater than 0.8 and quality scores. The figures below show that the generator is able to climb the different scores. We also checked the distribution of checker values to ensure that the defined values were respected. We also compared the amino acid distribution from the initial dataset of active peptides and the final dataset of generated peptides. We can see that the distribution is very similar and follow the same trend, meaning again that the generator learned the syntax of the active peptides. To help analyze the output of our generation, we also added some tools in our pipeline. We can make a clustering of the generated peptides as illustrated in the figure on the right. A UMAP clustering was made with 100 clusters. Each dot represents a peptide and each color represents a cluster. This clustering is notably helpful to make a final selection among the generated peptide. We recovered from each cluster the one peptide, maximizing the scores. This helps gaining in diversity in the final selection among the generated peptides, while preserving the best scored. Also, we incorporated to our pipeline PepsA, a tool developed by Merck, that allows the visualization and alignment of peptide, including non-natural amino acids and non-linear peptides. This tool can be really useful to help the biologists and chemists to analyze the final selection of generated peptides. For instance, the figure on the left represents the alignment of some generated peptides from a single cluster. We can see that the peptides from this same cluster share a big residue sequence pattern, validating the similarity inside clusters. Thank you for watching our video. If you have any questions, please reach out, we'll be happy to help.